Now, let's look at mass flow, mass transport in the phloem. And let's define what a source and sink is. So the source is a little bit more straightforward, I would say, because it's basically where the assimilates are being produced, which is where the leaf, really. The leaves are sources. Remember from the structure of the leaf, from the cross-section across the leaf, that you've got your palisade mesophyll cells, which of course do photosynthesis. And then there's the vascular bundle inside the leaf, right? So the assimilates are made in the palisade mesophyll cells, and then they're loaded into the phloem. So the source is always the leaf. Now, what are the sinks, though? The sinks are the location where the assimilates are used and or stored. So basically, they're taken out of the phloem and they're put into the sinks. What are some examples of sinks? Well, the roots. Let me just change color to make it a bit more obvious. The roots, for one. Right? The root is a sink. Doesn't have access to sunlight, cannot photosynthesize, has to rely on a supply of nutrients, of assimilates from mass transport through the phloem. Often the roots might have something like a potato tuber being formed. Potato or tuber, they're called collectively, like the root vegetables, which will store a big amount of sucrose that gets converted into starch, right? So that is also an important sink. A growing flower can also be a sink because it needs a lot of nutrients to develop. And so the movement of assimilates from source to sink is basically what we define as translocation. Quick mention of a possible trap that the potato tuber, for example, depending on the stage in the life of the plant, it can act as a sink, but if you take a plant, or a seed can act as a sink, but if you take a plant and plant it, so if you just like plant a potato that sprouts and makes roots and stem, at that stage the potato can be a source. Because the plant is still a seed, it can't photosynthesize. Don't worry too much about that. That it might be like, it's rare. It might be a sort of tricky question, but focus on the basics. Okay, so what are the stages of mass flow? Basically, of mass flow, of translocation. So the word mass flow and the word translocation are not mutually exclusive. Sorry, they're not interchangeable, I meant to say. The word translocation, as we said, is simply the flow of assimilates from source to sink. Mass flow is a much bigger, sort of broader process that involves both the phloem and the xylem together. And it's a theory or a process that was described by scientists relatively recently. So let's break it down into steps. So step number one, I'm just going to do them in slightly different colors here so you guys can. Step number one, the loading of assimilates into the phloem at source, which we already saw. It's all that stuff that we said with the hydrogen ions and the co-transport. That leads to sucrose or amino acids as well, right? Being loaded into the sieve tube elements here. So here is my loading of my assimilates, number one here, into the sieve tube element. And what is the effect? What is the impact of this happening? It lowers the water potential, which we can sometimes symbolize like that with the Greek letter psi. Why? Whenever we have the addition of a solute, that will lower the water potential. Remember, concentration of solute and water potential are opposite to each other. So that lowers the water potential in here. So what does lower potential do? What is going to be the consequence of that? So number two, the consequence of that is that if you've got low water potential in here, you're going to tend to want to draw water in. Where is this water going to come from? It's going to come from the xylem. That's the close. the closest source of water here. So here, this is the xylem. This is my phloem, my seep tube elements, right? Xylem is transporting water up from roots to leaves. So number 2 is that water is going to be drawn in from the xylem and it enters the phloem by osmosis, as always. Water moving is always osmosis. Thereby increasing, so what is the effect now of water being drawn in? More fluid. It increases the hydrostatic pressure. Again, this idea of hydrostatic pressure, like we saw with the blood vessels. More fluid, more pressure of the fluid against its container. So it increases the hydrostatic pressure. In here, I hope you can see that HP. It increases the hydrostatic pressure. Step number 3, let me just change color. Step number 3 is going to be that so now you have an increase of hydrostatic pressure up here, which will basically push, it's going to create a net push of the water plus assimilates the sap basically downwards. So there's going to be a big push downwards. When the assimilates now reach a region which has, and of course, here is my source, right? And here's my sink, which is typically the root. When it reaches here near the region of a sink, the assimilates are going to be unloaded, into the sink. So number 3 here is that the assimilates are loaded into the sink. What is that going to do? It decreases the water potential in the phloem. So therefore, some water, let me just do yet another color. Let's do purple. So water is also going to leave the phloem. So number 4, water is also going to leave the phloem and go into the sink. Why? Because when the assimilates got moved into the sink, got unloaded into the sink, what did that do for the sink? It lowered its water potential. So water needs to be drawn in. Where is that water going to come from? It's going to come from the phloem again. So now the hydrostatic pressure, number 4, now the hydrostatic pressure, let me just do purple again, sorry. The hydrostatic pressure now is decreased. Compare that to that. And finally, this entire flow creates a hydrostatic pressure difference or gradient, you could also say, which pushes sap to the location that is required by mass flow. So basically this here is my, let me just choose another color. Let's do orange. Hopefully those are different enough. So step number 5 basically refers to this net hydrostatic pressure that is created. Just going to give you like 2 seconds to copy down the annotation on the diagram. If you can use different colors, try. If you don't have them handy to yourselves now, just kind of like correlate the numbers to the part of the diagram, and then later you can fill it in with colors. Take a picture of the screen if you want. It's probably faster. And we are now going to move to a example question. Plants have developed a mechanism to ensure that assimilates are transported to where they are most needed. Describe how assimilates are transported through the plant. So 4 marks. So our context here is the translocation, the phloem and mass flow. Our command word is describe. And our guide, a direction, is that we are dealing with only assimilates and not water. Take a minute, just a quick minute to try to structure the answer for yourselves. So again, this is one of the questions with what we tend to call a generous mark scheme, which basically means that there are more marks available to mention than the 4 marks that are needed in the question. Let's go through them. So again, it's a describe. We don't need to give a lot of reasons for it. We just need to be as detailed as we can, making sure that we cover at least 4 marks. Just a super quick advice that especially if you're going, for really, really high marks, it's often not a bad idea to write, like, don't just say, oh, I wrote 4 marks, fine, I can stop now. Try to aim for writing at least 1 more mark, because you never know, sometimes you might have not phrased something exactly as it should be. So having that extra mark there is going to ensure that you're not going to lose any marks. So assimilates are loaded into the sieve tube elements of the phloem at the source, mark number 1. Just do it right, so it's a bit more obvious. What does that do? What is the effect of that? It decreases the water potential of the sieve tube elements and decreases the water potential. That's 1 mark. And what's the consequence of that? Water from the xylem moves into the phloem by osmosis. That's another mark. Thereby, what's the result of that? Increases the hydrostatic pressure of the phloem at the source. It's really important to, state the places where everything happens, to be precise. Now, at the sink, the assimilates are unloaded and water follows, thereby decreasing the hydrostatic pressure. The fact that I've got increased hydrostatic pressure at the source and decreased hydrostatic pressure at the sink, means that this difference in pressure between source and sink transports assimilates by mass flow. This difference here also gets a mark. So it's basically the net difference in pressure and hydrostatic pressure between source and sink that drives mass flow.